If you're preparing for Lab Practical 2, Lab Practical 2 is basically the respiratory system and the digestive system. And as I mentioned before, and the purpose of the lab is to identify the different structures, different layers, and so on, which is the labeling part, which is the major part that you need to know for the lab practical. And this is what we should be learning during the lab, labeling. What is this and what is that? And this is a major part, but also there should be or we should ask you some questions that I call applied questions, like do you understand or you just can label? Do you know, like this is the duodenum, this is the pyloric sphincter, this is this and that. That's part or the major part. But do you also know how to apply your knowledge and and intermingle like the information that you understood from the lecture, which is basically physiology, with the anatomy or not? So beside labeling, which is your part, that's what you need to learn during the lab. And you also can go to the learning center or the bone room and learn more about the labeling. But as you're labeling, you will need to ask yourself some questions. Not only what is this and what is that, but you have to know the general functions of different parts. So as you go, you will need to ask yourself, what is this? This is the bronchioles. Why do we have it? What's the function of? So Lab Practical 2 AP2 Lab Practical 2 is respiratory system and digestive system, as I mentioned. So what else beside labeling? You can be asked about the general functions of different parts, and this is what I was just talking about. Plus oxygenated, deoxygenated, specifically the pulmonary arteries and pulmonary veins. And the rule of the thumb in this part specifically is every red vessel is artery, every blue vessel is vein, except pulmonary circulation is the opposite. The other thing is routes, sequences, like the bronchial tree. Not only the bronchial tree, but also the pulmonary vessels. Where is it going to? From which part to which part? So this is for the respiratory system. For the digestive system, same thing basically. What's the general function of? Why do we have this? Why do we have that? Plus routes and sequences, like the biliary system, for example, like the portal, our hepatic portal circulation, where it goes from what to what, and so on. So general functions and routes and sequences, and if you see numbers, that will be important as well. So I'm going to go through some pictures showing you the respiratory and digestive system, and I will mention a little bit about each one of those, whatever applied question that can come to my mind, general question, general function, routes and sequences as we go with both the respiratory and digestive system. So if we start with this model, for example, generally speaking, the routes here are going from the nose to the ventricle, passing through the meatus, which is in between the conchi, and then going to the posterior part, going to the pharynx. If we wanted to learn a little bit more about the applied questions here, let's say something like the frontal sinus and the sphenoid sinus and the ethmoid air cells here. Why do we have it? We have it, generally speaking, this is not physiology, this is anatomy, but also we need to know a little bit about it. So why do we have the sinuses? To make the skull a little bit lighter weight for the body. So this make spaces to make the skull bone weight less or not heavy, less weight, and to resonate the sound as well. So this is for the sinuses. Something like why do we have the meatuses? We have the meatuses so that the air is forced to go through them. So as it is going through in between these tiny tunnels or tiny cavities or tiny passages, it will be humidified. The dust or anything there will be trapped. So you're cleaning the air. You are humidifying the air, and you are making the air a little bit warmer. The perfect air for your lung is the air that's clean, the air that's warm and humidified. So this is the best. How to do that? go through the nose, go through the meatus. So this is a function of the meatus. If you go like to something like the opening of the auditory tube or nasal or pharyngotympanic tube or eustachian tube, why do we have it? To equilibrate the pressure between the either side of the eardrums. So if you see it in this model, the opening of the eustachian tube, or that will be the opening that alleviate or equate the pressure on either side of the eardrums. Something like this, the adenoids or the nasopharyngeal or pharyngeal, which has three names. Tonsils. Why do we have the tonsils in the first place? This is for defense mechanism. Again, I'm not explaining. I'm not going in depth with the physiology, but just something related to the anatomy, which is applied parts. So why do we have any one of these tonsils? Defense, immunity. If you go to something like the hard palate, soft palate, and the uvula, the palate in general is separating the mouth from the nose. Why do we have the uvula? The uvula can open and close to prevent any solids, any food, any drinks. When we eat it, go from the mouth. It will have three routes. It can go up to the nose, and this can be prevented by the presence of the uvula that open and close, or it can go down. So it can go up, prevented by the uvula, or go down. Now, if it is going down, it can go to the air, which is the larynx, and it can go to the esophagus. That will be the function of the epiglottis that we will see in next slides. So if you look at this slide, if you're moving down, the food enters from the mouth and it goes down. It can go up to the nose. That's prevented by the uvula. And if it goes down, it can be directed to the larynx or the esophagus. If you're eating or drinking, you need to close the route that's moving toward the larynx, and this will be the function of the epiglottis. So why do we have the epiglottis? To protect the respiratory passageways, or to prevent the foods or drinks from entering to the lung. This picture containing what I was just talking about. So when you eat, the food goes in the mouth and goes to the oropharynx. And as we know, the pharynx is three parts. The upper part, which is behind the nose, is called nasopharynx. The middle part, which is behind the mouth or behind the oral cavity, is called the oropharynx. And the third part, which is the lower part, is behind the larynx, and this is called the laryngopharynx. So we have three parts. What's the function of each one of those? The nasopharynx, beside containing the nasopharyngeal tonsils, it's also the route for the air to go through and enter to the oropharynx. What's the oropharynx? The oropharynx is a common pouch or a common cavity for both the digestive and respiratory tract. So the air coming from the nose go to the oropharynx, which is the middle part. The food and drink coming to the oropharynx, coming from the mouth, will go to the oropharynx. So the oropharynx is a common bowel or a common cavity for both respiratory and digestive system. And then you will go down. If this is the air, you want it to enter to the larynx, to the trachea. If this is food, you want it to go to the laryngopharynx and then go to the esophagus. How to direct that? Again, if the food or drink is entering to the mouth, it goes to the oropharynx. You need to prevent the food from going up, and this is a function of the uvula. The food will go down, but it has two routes. It can go to the larynx, or it can go to the laryngopharynx. In this case, the larynx, when you're eating, the larynx move up to be closed by the epiglottis, and this is actually the function of the epiglottis. So beside understanding that this is the epiglottis, why do we have it? Always ask yourself, why do we have it? Why do we have this? Why do we have that? And so on. So why do we have the epiglottis? Is to protect the airways, the food from entering the larynx and trachea and the respiratory system in general, because it will go all the way to the lung. So this is the function of the epiglottis. If you are in the airway, you see the vocal cords. Why do we have the vocal cord? The vocal cord is for production of the sounds. It's open and close for the air to go in and out, to and out of the larynx. So why do we have the vocal cords? Producing sound, and by opening and closing the passageway to the air to and out of the larynx. So this is for the epiglottis, for example. As you move down, we know that the light blue structures are cartilages, and generally speaking, in 234 lab, usually we do the way the models usually colored is light blue is usually, of course that's not a rule, but most of the time the cartilage is light blue. The bone will be like off-white or beige. If you're talking about the intestine, we'll talk about that later, but there will be different colors that we will talk about, and it can help a little bit, but we should know without this, of course. This is just a hint. But generally speaking, if you're going to the larynx, you have laryngeal cartilages. Why do we have it? Same thing. We have one thyroid, which had the thyroid prominence, or Adam's apple, and then you have the cricoid cartilage, and then you have many paired and single cartilages. Why do we have it, the cartilage? To keep the larynx open, to protect the larynx and keep it opened. Why do we have the other tiny ones? To help the production of the sound, just the general function of. If you go down to the esophagus will have the, the trachea. If you're going to the trachea will have the tracheal cartilages, or called the C cartilages, C rings. Why do we have the C rings, and why is it C? We have it to keep the trachea patent or opened all the time, so it does not collapse or something if you're sleeping. So that's why we keep it open. Why is it C-shaped? It's C-shaped, and there is a muscle in the back end membrane, and this is because if you're swallowing, the food should move down the esophagus, and there will be a space for it to move smoothly. If this is a complete cartilage, you will have some sort of dysphagia, or the food will be hard to swallow as it moves down the esophagus because there is no room to expand. But we have it's C ring that's not continuous from the posterior aspect. It contains a membrane there to allow the expansion of the esophagus when the food is moving down, so it moves down smoothly. For this one, we have the hyoid bone, we have the thyroid membrane, we have the thyroid cartilage. The tip of the thyroid cartilage that's prominent anteriorly, and it's more obvious in men or males versus females. So this is called the prominence, laryngeal prominence, the prominent part anterior of the thyroid cartilage. The thyroid cartilage is also called Adam's apple. So this is why we have these cartilages. If you look at the thyroid gland, why do you have the thyroid gland? To produce thyroid hormone. No details, but the general function that's applied to what we're talking about in the lab. For this model, if you follow down with the trachea, it will take you to the rest of the bronchial tree, but it's important in this part to realize that if you're looking at the red blood vessels, that will be obviously pulmonary veins, and this is a very common mistake that we automatically say anything red is artery. No, anything red is oxygenated. Not necessarily. Most likely it's an artery, but this is an exception. So if you're looking at this picture and you're seeing the red blood vessels, that will be the pulmonary veins. If you're talking about the blue, that will be the pulmonary arteries. And if you look at these tiny ends of the bronchial tree, this will be the tertiary and the secondary. Contains secondary and tertiary, both of them in the same picture. But the tips, the last part of the tree will be the tertiary. This is what we can see here, and it's also important to know the direction of the blood. So in this part, you need to know that red is vein, pulmonary veins. The blue is pulmonary arteries. This is one thing. The other thing is what's the direction? Like the blue is taking the blood starting from the right ventricle, which is deoxygenated, go to the pulmonary trunk, go to the pulmonary arteries, right and left, and then it will divide. So the direction is from the right ventricle to the pulmonary trunk to the lung, which is carrying deoxygenated blood. If you look at the red, on the other hand, this is the opposite. It contains oxygenated blood after being oxygenated in the lung, and then the direction is it's carrying the oxygenated blood from the lung going all the way until it reaches the left atrium. So it's important to know red is vein, blue is artery. That's one. Direction of blood, and is it oxygenated or deoxygenated. If you go down here to the diaphragm, why do we have the diaphragm? The diaphragm separates the thoracic cavity from the abdominal cavity, and it moves up and down to help with the respiration or breathing. And there is a difference between breathing and respiration that we need to know. That breathing is the inspiration and expiration. When you breathe in and out, this is called breathing. The respiration is the crossing of oxygen and carbon dioxide through different membranes. And there is as we learned in the lecture, there is external respiration and internal respiration. The external respiration is in the lung, which is in the alveoli, where the oxygen goes out to the alveoli and go to the external atmosphere or the external air, and the carbon, the carbon dioxide move to the alveoli and goes all the way to the outside, and the oxygen goes in. If you go to the cells, on the other hand, which is called the internal respiration, and again, respiration is moving in and out of oxygen, carbon dioxide in different directions. So if you're going to the internal respiration, which is the respiration of the cells of the body, anywhere, that will be the oxygen going in, and the carbon dioxide will be moving out. So function of each part and the routes. It's also important to know that the right lung contains 3 lobes, and the left lung contains 2 lobes. And the reason why is that the heart occupies the middle lobe of the left. So in the left side we have 2 because the heart occupied the middle. So on the right side we have upper, middle, lower, or superior, middle, inferior. It's the same thing. So we have 3 lobes, 2 fissures. On the left side we have 2 lobes and 1 fissure only. So the difference is 2 in the left side, 3 in the right side, and where the heart is pushing on the lung, this will create a notch that's called the cardiac notch, which is the notch or the curvature that's produced by the heart pushing the lung. If you look at this picture, you will see the trachea and the bifurcation of the trachea into 2 primary or main bronchi. This is called the carina, right in the middle. This is where the bifurcation occur. And now the numbers are important. We have 2 primary or main bronchi. Each one of those correspond to the number of lungs. We have 1 right lung, 1 left lung. That's why we have 1 right primary or main bronchus, and we have 1 primary or left main bronchus, because we have 1 right lung, 1 left lung. If you go to the secondary bronchi, and it's also important to know the names of each one of these structures. There are more than 1 name for each one of those. Like primary is called main. Secondary, we have 3 secondary bronchi in the right side, and we have 2 secondary bronchi on the left side. Why? Because the secondary bronchi are also called the lobar bronchi. So secondary or lobar bronchi. Why? We have 3 on the right and 2 to the left, because it's lobar, so each one of these will go to the lobe, and we have 3 lobes in the right side, we have 2 lobes in the left side, so the number of the secondary bronchi correspond to the number of lobes. If you go to the tertiary bronchi, which are all these colored terminal parts of this bronchial tree structure or model, that will the number correspond to the number of segments or lobules, and this will be that's why the tertiary bronchi are called the segmental, because each one of those will go to a segment where it's called the bronchopulmonary segment. So it's the tertiary going to the segments. So 2 names for each one of those, and where is it going to, and this will correspond to the numbers. So if you're talking about the numbers of tertiary bronchi, also known as segmental, this in the right side we have like 10, and the left side we have 8 or 9. Why? Because we have 10 segments in the right, and the left might have like 8 or 9. It's different from person to person, so the number corresponds to something, and this is important to know as applied parts of learning the lab. The passageway is also important to know, like if you're breathing, the air will go from the nose going to the nasopharynx, to the oropharynx, to the larynx, to the trachea, to the primary bronchi, secondary bronchi, tertiary bronchi, and then bronchioles, terminal bronchioles, respiratory bronchioles, alveolar duct, and then to the alveoli. So this is important to know as applied part or applied knowledge that's related to the lab, that if you're learning, you need to learn also the directions and the routes. For the digestive system, we will do the same thing. So general functions, just generally speaking, the simple knowledge about the function and the route and sequences of different parts. This is a picture that's showing the different parts of the digestive system, digestive tract and accessories, and there is a difference between those. The digestive tract is the tract through which the food will move when it enters the mouth, going all the way down, which is the route until the remnants exit out of the anus. So you should know the sequence starting from the mouth, going to the oropharynx, to the laryngopharynx, to the esophagus, to the stomach, to the small intestine, duodenum, jejunum, ileum, and then to the different parts of the large intestine, starting from the cecum, ascending, transverse, descending, sigmoid, rectum, anus, and so on. The function of each one of those is just a simple way. Esophagus, this is a tube that delivers the food from the mouth to the stomach. Why do we have the stomach? Mixing and breaking down the food. Small intestine, it's basically for digestion and absorption. Large intestine, why do we have it? This is for absorption of water, and also production of vitamins and reabsorption of other things, including the bile, but it's more important. There are a lot of functions, but I'm talking about the general functions. So here it's reabsorption of water and production of vitamins. Most important thing about the large intestine. If you look at the accessory structures, what's the main function of the liver? There are a lot of functions. Defense, metabolic function, but one of the functions related to other structures is production of bile. Where do we produce the bile? Where do we synthesize the bile? It's in the liver, not the gallbladder. So why do we have the gallbladder? The gallbladder is for storage, even though it modifies a little bit of the bile, but it's not produced in the gallbladder. This is a common mistake. It's produced actually in the liver. So bile is produced from the liver, bile is stored in the gallbladder. So what's the main function of the gallbladder? Storage of bile. How about the pancreas? The pancreas is important for secretions. What are the secretions? Different secretions. We have exocrine secretions, which are the digestive enzymes. Endocrine secretions, like the islets of Langerhans or the islets of the pancreas, produce insulin and glucagon, which is endocrine function. The third function is production of buffer, and the buffer is to neutralize the acidity as it goes to the duodenum, because as we know, the pancreas will open to the duodenum, so the food coming from the stomach is very acidic, because we know that stomach produces the stomach acid, and this is one of the functions of the stomach too. So it's mixing, digesting, and helping breaking down the foods and killing any pathogens in there. And the function of the stomach acids, or the hydrochloric acid that's produced in the stomach, is to break down the proteins, break down different foods, and sterilize or clean the foods from different pathogens that might be in the food, because we know that the food is not obviously sterilized or very clean or something. It might contain pathogens, and that will be killed by the stomach acid. So as the food moves out of the stomach, it's very acidic, so the pancreas will produce some bicarbonate or some buffer to neutralize the acidity, which is also one of the functions of the bile. Why do we have the bile? The bile is for emulsification of fats. So when you emulsify the fat, you make it small pieces so that the enzymes can break it down. With that, the bile will also contain some buffer system as well, or buffering of the acidity. So how do we buffer the food that's coming from the stomach to the duodenum? It's buffered by two things: by the pancreatic secretion, and the pancreatic secrete exocrine, endocrine, and buffer. Exocrine is the enzymes, endocrine are the hormones, glucagon and insulin, and buffer is the bicarb. Why do we have the bile? Emulsification of fats to help digestion of fat, and also contain buffer, which is the bicarb. That's why as the food moved out the duodenum, it will be buffered, and the acidity will be pretty much neutralized. As we move down the digestive system, we will see many sphincters, and each sphincter will have a function that you need to know the basics only. So, like the first one is the cardiac sphincter, which is the sphincter between the end of the esophagus and the beginning of the stomach. Why do we have it? To prevent the food from moving up from the stomach to the esophagus, because the food in the stomach, it's not supposed to go back. The food should go from the esophagus to the stomach, and it shouldn't go back. It is acidified. It's too dangerous to go back to the esophagus. It will burn it. So we have the cardiac sphincter, or the lower esophageal sphincter too, because to prevent the food from going back to the esophagus, burning the esophagus. If the food moves down, it goes to the pylorus, and there is a muscle that guard the opening of the stomach to the duodenum. So the food, after staying for some time in the stomach, it should move down to the duodenum. What's the name of the sphincter here? It's called the pyloric sphincter. So what's the function of the pyloric sphincter? Keep the food in the stomach until it is digested, and then it will open so that the food moves from the stomach to the duodenum. So now the food going from the duodenum to the jejunum to the ileum, and then it will go to the cecum. There is another valve here, or a sphincter, that's called the Luc Kell Valve. Why do we have it? To prevent the food from moving to the ileum to this until the cecum, until it's the right time after after absorption, and then the food will go to the large intestine. There is the appendix here, or the veriform appendix. The appendix here have nothing to do with the digestive or absorption function. It's actually part of its modified lymphoid tissue, and it is there to protect this area, and that's why we have the appendix in this area here. The food will move down the large intestine, and the direction again is important. So we start with the cecum, ascending, right flexure, transverse, descending, sigmoid, colon, rectum, and then you go to the anus. In the anus we have two sphincters, the internal and the external. The rule of the thumb is always the internal is involuntary. It is automated. We don't really control it, but the external one is voluntary. So and these are the different sphincters of the anus, and this will keep the remnants or the feces within the rectum and the anus until it's the right time. And then the inner one will open by distension only. So once the rectum and the anus started to distend, the inner one will open, but the external one will not open until you voluntarily relax it. If you look to this picture here, you will see different parts of the pancreas, and you'll see the pancreatic duct, the main one, and the accessory pancreatic duct. What's the function of the pancreatic duct? It takes the secretions, which are, as we named it before, the exocrine, which are the enzymes, digestive enzymes, plus the buffer. Obviously, the endocrine part does not go to the duct, because endocrine means ductless. It will go directly to the blood. So what moved, or what's the function of the pancreatic duct? It's to deliver the pancreatic enzymes, digestive enzymes, and also the buffer to send it to the duodenum. And as we know, the common bile ducts should join the main pancreatic duct. So the main pancreatic duct plus joining the common bile duct will open to the major duodenal papilla, which is the inferior one. The superior one is for the accessory pancreatic duct. So if I ask you, why do we have the major duodenal papilla? This is for opening of secretions coming from the common bile duct and from the main pancreatic duct. What are these secretions? Coming from the pancreatic duct, that will be the digestive enzymes and the buffer. What is coming from the common bile duct? That will be the bile and also buffer. So all of these will go to the major duodenal papilla, going to the stomach. I'm sorry, to the duodenum. How about the minor papilla. Why do we have it? This is for the opening of the accessory pancreatic duct. In case of the main one is closed or something, this will open. This is still open and go to the minor duodenal papilla, and it's going to open to the duodenum. If you look at this picture here, this is a model that's showing you the intestinal layers, and there is a difference when you say layers versus different parts. So there is a mucosa, submucosa, the muscularis, musculosa, or the musculosa, and then the last part, or the outer part, is called the serosa. The question will be, why do we have these? Like, here is the mucosa, which is the outer part. It has the villi. Ask yourself, why do we have villi? We have villi to increase the surface for absorption of food. So if you unfold all these villi, it will make it a long, very long surface, or very huge surface for absorption. So the villi are there for increasing surface for absorption. If you look inside the alveoli, you will see lacteals, which is part of the lymphatic system, but this is a specialized part that's not there for the immune system. It's basically here for absorption of fat. So when we absorb carbohydrates and proteins, this will be absorbed the regular way from the villi, going from through the hepatic portal system, going to the liver, not the fat. The fat will go to the lacteals. And from the lacteals, and the lacteals are equivalent to the systemic capillaries. So from the lacteals, which is in the villi, to the minor lymphatic vessels, going to the other vessels. And in this model, anything that's light green is part of the lymphatic system. So it will move down until it goes to the smaller lymphatics vessels, large lymphatic vessels, and it will move on and on until it go to the systemic veins, go to the systemic bloodstream, and it will go to the liver anyway, but indirectly. So what's the function of the lacteals? Help absorption of fat. You will see something like a lymphoid nodule in here, and this is the part that's actually for defense, which is the big green round structure here. In the submucosa, you will have blood vessels. Arteries are red, of course, and blue are veins. And again, any red is artery, any blue is vein, any green is usually depending on which part. It can be the biliary system if we're talking about the liver and the biliary tract. In this light green, that would be the lymphatic. But generally speaking, if you're talking about the arteries and veins specifically, the veins are always blue, the arteries are always red, exception of the pulmonary and the fetal circulation, but we're not talking about it right now. So for this part, if you're talking about specifically respiratory and digestive and the rest of the body, only the pulmonary system will have them the opposite way. Now the next part will be the circular and longitudinal muscles, and the way here to recognize those is the location, not how they look like, because this is a cut section that shouldn't tell you much. It depends on the orientation. How are you looking at it? So the inner is circular and the outer is always longitudinal. This is a rule of the thumb. Circular muscles are always internal and the longitudinal muscles are always external. So the muscle layer that's closer to the serosa, that will be the longitudinal. The inner muscles that is closer to the submucosa, that will be the circular muscles, and this is your only way to recognize, not how they look like, because you're looking at a cut section which doesn't tell you much. It depends on the orientation. So don't count on that. Count on circular is internal, longitudinal is always external. In this model, you can see also some white structures that are called the myenteric. It's called the myenteric plexus, which is controlling. This is the nerve control of the intestine. In this model here, we will need to know the basics: right lobe, left lobe, and there's also caudate and quadrate lobe. The caudate means, cauda means tail, so this is the lobe that has some tail, and the quadrate lobe is the lobe that looks like a quadrum, or that have 4 different borders. And the caudate is beside the inferior vena cava, the quadrate is beside the gallbladder. Why do we have the gallbladder again? For storage of bile. There is one door for all these structures to enter and exit out of the liver, and this is called the porta hepatis. In the lung, we call it the hilum of the lung. The hilum of the lung is where things enter and exit, and this is important to know the direction. Are you going entering to the lung or exiting of the lung? If you're entering to the lung, that will be arteries. If you're exiting, that will be veins. And generally speaking, any door or any porta or any hilum of any organ, it will contain the basics, the main things, plus or minus other things. But you will always see an artery, a vein, lymphatic vessel, and nerve entering and leaving. So in this case, the hepatic artery will be entering. The hepatic portal vein will be also entering, even though it's the vein. But the hepatic portal vein is a vein that carry, and this will be the function, the related function, that's carrying the blood containing absorbed nutrients from the digestive system, bring it to the liver, and end at this. This is the only function. So it is entering. While the hepatic vein will go to the inferior vena cava, so it's exiting. So what's entering, what's exiting? The artery, hepatic artery is entering. Hepatic portal vein or portal vein will be entering as well, just like the arteries, while the hepatic duct will be exiting, carrying the bile out of the liver that's been synthesized in the liver. So porta hepatis is this porta or this door of the liver that contain biliary tree, the veins and arteries. And it will also, but it's not shown here, lymphatics and nerve supply. And this is the rule in everywhere basically. If you have the door, which is called hilum, hilus, or porta, it doesn't matter. This is basically for entering and exiting. And again, you need to focus on what is entering and what's exiting. It's not just names. You need to know the direction of each one of those. Are you bringing blood to the liver or are you leaving the liver? When you look at this picture, this is showing you the liver, but it's kind of removed part, a section that was removed to show you what's inside. And basically all these colors are, we have red, blue, purple, green. The red are branches of the hepatic artery. The purple are branches of the hepatic portal vein. The blue are branches of the hepatic vein, which is obviously different than the hepatic portal. And the name here is portal, so you call it portal vein. Hepatic portal vein, but you have to put portal anyway. So if you're describing the veins, the venous system that take the blood containing absorbed nutrients from the digestive system, bring it to the liver. So obviously the route is entering, and this will be the hepatic portal vein or portal vein. While the branches of the hepatic vein, you don't put portal now, are leaving, and it will bring the blood leaving the liver to the inferior vena cava, and there will be right and left hepatic veins leaving, and this is the blue. The green, this will be the biliary tree, and you need to recognize the different parts that participate, which is right hepatic, left hepatic duct joined together to make the common hepatic duct that will join the cystic duct, which is coming from the gallbladder, making the common bile duct. So again, red, branches of the hepatic artery, blue, branches of hepatic vein, purple, branches of the hepatic portal vein, and green are the biliary tree. Now the salivary glands. We have parotid glands, which is outside of the masseter muscle under the ear, and you have the submandibular tonsils. Salivary glands. And you have the sublingual salivary glands. Generally speaking, why do we have salivary glands? To produce saliva. What's the saliva? It's a fluid that's important for moisturizing the food, and it also produces some enzymes to help breaking down or just starting the breakdown of the food. And these enzymes are lingual lipase and salivary amylase. So the saliva contains salivary amylase. So what's the function of the saliva? Producing mucus secretion that moistening the food, moistening the mouth, and salivary amylase, which is the enzyme that breaks down amyl, which is a starch or carbohydrate. If you're talking about the mouth in general, there is a lingual lipase produced from the tongue, and the lingual lipase is from the name lipid. This is to start the process of breaking down of the lipids. When we talk about the teeth, it's important to, of course, labeling is one thing, but it's important to know the teeth from each other. So you need to count, like start from the central incisor, the lateral incisor, the canine or cusped, and then you have the first premolar, the second premolar, the first molar, the second molar. There is no third molar here, and the third molar usually start to show up somewhere between 17 and 21. So if this is obviously this picture is for a kid or a teenager that's younger than 17, 18. So this is basically a child or a kid or teenager or something. It's not an adult. Why? How did I know? Because it does not contain the third molar. So counting and knowing the age is important here. So third molar, which is a wisdom teeth, will show up somewhere between 17 and 21. So if you see 1, 2, 3, 4, 5, 6, 7, this is 7. So another 7 there, that will be 14. So this is obviously somebody who's younger than 17. You're talking about a child or teenager or something that's younger than 17. If this was an adult, you will see the third molar, which is the wisdom teeth, and if that's the case, you will have 8, another 8 on the other side. That will make it 16 lower teeth, and that will be 16 upper teeth. So numbers here are important. How many teeth we have in the upper jaw and in the lower jaw, the upper row of teeth and the lower row of teeth. If I ask you this, you will need to ask me what's the age. If I'm saying younger than 17, a child, a teenage, you will say we have 14 and 14, so the total is 28. If I'm saying this is an adult, so you have 16 upper and 16 lower, so that's together will be 32. So it's important to know the numbers here. And if I ask you the upper row, which is the same number as the lower row, you have an adult, 16 upper, 16 lower, that will be 32. If we're talking about younger than 17, that will be 14 upper, 14 lower, so 28. That will be the total. And if you're talking about the teeth, the inner structure of the teeth, it's important to differentiate between the different parts and the different layers. This is something completely different, and you don't need to be confused about this. If I'm talking about the different parts, then I'm talking about this part up here, the pink, which will be this part. I'm not talking about the layer, the part that will be the crown. The part in the middle will be the neck, and the rest will be the root. If I'm talking about the layers, this is something different. So the outermost layer, that will be the enamel. The next layer will be the dentine, and then the cavity. And the cavity is pulp cavity, which is inside the pulp, and the root canal. So the inside is a canal in the root and a cavity in the pulp. So again, you need to read the question twice before you answer. Always read the question and see what is the question. Don't just jump and see the sticker and or where are we pointing to and then just answer. No, that's not the way to answer. The way is what's the question? Are you asking about layers or are you asking about parts? Parts, crown, neck, root. Layers, enamel, dentine, cavity. What's the name of the cavity? The cavity in the upper part, the crown part, the upper part here, that would be called the pulp cavity. If you're talking about the cavity or the canal within the root, that would be the root canal. And the root canal will contain the blood vessels and nerves that's entering and leaving. And again, the direction is important. The artery is entering, the nerve is leaving at this point because it's sensory, and the vein will be leaving. So and the lower part here will be the apical foramen. Why do we have the apical foramen? For entrance and exiting of the blood vessels, which are the arteries and veins, and the nerves. You also have cementum, which is the white that's right surrounding the roots and the neck, and this is to cement or to fix the tooth to the gum and the bone. And you also have the periodontal ligaments, which are these transverse ligaments, again to fix the teeth in place. So always ask yourself, why do we have this? Why do we have that? That will be the wise way to prepare for the lab practical. One more thing I wanted to mention here is always do one at a time. So if I'm asking about three different things in this model, what is this? What is this? What is this? Don't say in your mind the answer is enamel, dentine, and gum, for example. No. What is this? This is enamel. This is question number one. Answer question number one. You're done. Go to the next. What is this? This will be the dentine. Okay, what's question is this? This is number two. Okay, question number two is dentine. And then what's three? This will be the gingiva. Okay, answer it. If you answer the three questions in your mind and you think you will write them the right way, and you just sort them out the wrong way, or while you're answering you do dentine, enamel, gingiva, that's enough to give you two wrong questions. Or gingiva, dentine, enamel, you get everything wrong. So you need to do one at a time. This is my advice in the exam. Read the question twice. Ask yourself the question: are we asking about part or layer or cavity or what? And one at a time.